Okay, let's get started. I was going to say how pleased I was to see all of you, but the lights are in my eyes and I really can't see very well. But welcome to the 2013 Gruber Science Fellowship Lecture. For those of you who might not know me, I'm Carl Hashimoto. I'm professor of cell biology at the medical school and administrator of the Gruber Science Fellowship at the graduate school. The dean of the graduate school, Tom Pollard, unfortunately could not be here, so I'm filling in for him as best as I can. So I just want to say a few words about the Gruber Science Fellowship. It was established in 2011 with what Dean Pollard characterized as a breathtaking gift from Patricia and Peter Gruber for the support of students pursuing doctoral studies in the basic sciences. We have close to 70 Gruber Science Fellows now, and come this September, we're going to have 22 new fellows. And as Dean Pollard said, 100 years from now, when surely none of us are going to be stomping on this earth, there are going to be Gruber Science Fellows running around here, pushing back the frontiers of science, which I think vividly evokes the long-term impact of the Gruber's gift. I'm very pleased to say that Patricia Gruber is here with us today. Pat, would you mind standing up so everyone could see you? Everyone, please, join me. Thanks, Pat. So, to introduce our distinguished lecturer, we have called upon the services of one of the most distinguished scientists at Yale, Sterling Professor of Molecular Biophysics and Biochemistry, and 2009 Nobel Laureate in Chemistry, Tom Stites. Tom?
What a light. Well, it's a great pleasure to be able to introduce Bruce Alberts. Bruce was an undergraduate at Harvard and then a graduate student in biophysics, and we overlapped for a couple of years at Harvard. So that's where I first got to know Bruce. When he was working in the laboratory of Paul Doty. I would guess not very many of you are familiar with Paul Doty. He then went off to Geneva. In those days, you do a one-year postdoc. He did a terrific lab at Alfred Tissier's, and then went to Princeton, where he started his independent lab for 10 years at Princeton. And then after Princeton, he moved to UCSF, where he's sort of located there. He keeps, yeah, I think he has a house there, but he migrates around the world in recent decades. If I were to give a list of all the awards and prizes and honors and committees and things that he's been on, we'd be here for the next hour. But importantly, he was the president of the National Academy of Sciences from '93 to 2005, and he's currently, for the last 5 years, been the editor of Science. But I want to say a couple of things about the research that I know that he's done that has affected my research. And he started out working on DNA replication when he became independent and discovered something called single-strand DNA binding protein, and then started working on the process of replication, which I have to say is something that my lab has now been working on for the last couple of decades as well. But he found the proteins, many of the proteins, and participated in figuring out how they work. And one of the things I really found wonderful is his trombone model. You know, it's always good to have a good name for something. These days, everything's the XYZ protein, which interacts with the QXR protein, and I have no idea what it's about. But the trombone model was to try and explain how the replication system, the polymerase, can simultaneously go on both strands of DNA, because the synthesis only goes in one direction, but the DNA, of course, is going in opposite directions. So the idea is that what's called the lagging strand loops around, and the synthesis goes on in the lagging strand, and that loop gets smaller, and then it jumps to the next loop and gets bigger. So it's going in and out. And we were just discussing this. You sort of think you read something in a textbook, you know everything, but actually there's always just one more question. And so we're trying to ourselves figure out more details about how this all happens. He joined with others to write many versions or editions of the molecular biology of the cell, a very heavy textbook, I have to say. It's got everything you ever wanted to know in it. And he's been very important in promoting science, and particularly science education. I think that's what we're going to hear some about today, that one should teach science as a query rather than a list of facts. I mean, who cares about just listing those facts? You want to be able to answer questions, and I think that's what he promotes very effectively in his thoughts about education. So his title of his talk is Biology Past and Biology Future. Where have we been and where are we going? I always wonder where I'm going, I have to say. So, Bruce.
Well, thank you very much. It's a pleasure to be here. I'm sorry my friend Tom Pollard can't be here, but he's in my hometown in San Francisco at a dean's meeting. But I will talk. Just keep your eyes open. I have 2 slides with his name on it, and make sure you tell him that I did that. I can remember grading Tom Steitz's and Joan Steitz's exam papers in graduate school, so we go back a long way. I'm going to talk about some of the things that Tom introduced. He gave me a great introduction, because I'm going to start a little bit with talking about some things that I worked on, which, in order to make a general point about biology, which is the central role of protein machines in life, something we had no idea of when Tom and I were in graduate school. I'll then talk about the enormous amount that we don't know. As Tom and I were just discussing, unfortunately, textbooks make you think that we know everything because we write about what we know, but there's so much we don't know, and somehow we have to do a better job as textbook writers and making that clear, and we're hoping in our 6th edition, which we're in the middle of, we will do that. I'll talk about something that we also learned when we wrote the last edition. Every time we write an edition, we learn new things, and this idea of spatial organization inside cells, where everything's in the exact position. The cell is nothing like a test tube. And then going off topic a little bit, since I just came back from a major trip, I want to tell you about my life as a science envoy very briefly at the end. So I was a high school student when this picture was taken. Watson and Crick, 1953, made their famous prediction of how heredity worked based on the structure of DNA, double helical structure of DNA. Watson was only 25 years old. I use it to emphasize the importance of encouraging young scientists to do their own thing. And one of the very important features of the U.S. scientific systems that has made it so successful. Now the fact is, although Watson and Crick really solved the mystery, they knew nothing about the machinery that enabled a DNA sequence to be copied from one generation to the next. And the next breakthrough, just as I was in college, was the discovery of this enzyme called DNA polymerase that Tom works on and many other people work on. an amazing enzyme that will copy the DNA sequence in a DNA single strand by adding one nucleotide at a time based on the base pairing rules that Watson and Crick had discovered. And this is just the basic reaction from our textbook. The chain grows one unit at a time from the 3' end. The DNA polymerase only goes in one direction, a very important point, as Tom mentioned. And the energy for incorporation, making this work, it comes from the fact that the deoxyribonucleoside triphosphate is hydrolyzed every time you add a new monomer. Those two red phosphates leave. So this is where I started my own independent work in 1960 as a graduate student. And we all assumed, not only me, but a single enzyme alone replicated DNA. And the problem was, of course, that it would only work on a DNA single strand, and the Watson and Crick showed that DNA was double helical inside the cell. And moreover, it only went in one direction, as Tom was pointing out, and yet those two strands, as Watson and Crick discovered, are anti-parallel, go opposite directions. So I had a very dismal career as a graduate student at Harvard. Have you heard of Harvard? Me and many others were inspired by this theoretical success of Watson and Crick to make up theories about how everything works in biology. And I wrote a term paper for my first year graduate course about how DNA polymerase might be able to start its DNA synthesis on a chromosome even though the DNA was double helical. And I got an A+ on this paper, so I thought it must be right. So I then spent years trying to see if my theory was right. That was at least the first part of my graduate career. Of course, it wasn't right. There were no answers did not surprise anyone, and they did not add to the store of scientific knowledge. Nevertheless, after 5 years, my professor thought I had written up enough, done enough experiments, and all I had to do at Harvard was to go, what I thought was a pure formality, for a little oral discussion of my thesis with a set of famous Harvard professors. Nobody had ever failed at this stage in living memory. So I had already given up my apartment. I had a wife and a 1-year-old child, and my tickets to Geneva were bought for the next week. Little did I know, but I got a wake-up call. They didn't think I had done enough. They didn't believe what I wrote in my thesis. I failed my exam. This allowed me to write a whole paper about what you learn from failure. And in fact, older people like your professors or me have a certain wisdom because we failed so often. And people who are successful learn from their failures. So every time you fail, you can be a little bit excited about the fact that you're about to learn something. So the first thing I learned from my failure was the crucial role of strategy in science. Of course, I also learned that theoretical biology usually doesn't work because cells are too complicated, but having a good strategy in scientific research is the key to success. There are a million times more experiments you can do, especially now, than you should do, and so science is all about creativity and designing novel and effective strategies. So I did finally get my PhD. 6 months later, I did more experiments. I could then go to Geneva, Switzerland, and I suddenly discovered that the whole hypothesis that I had been working under was wrong. Work had been done on this large bacterial virus. Bacteriophage T4 has a long double-stranded DNA chromosome. Geneticists at Caltech had done an extensive analysis of how many genes there are in that bacteriophage and roughly what each one did. Just to remind you, this is a virus that attaches to the outer envelope of E. coli bacteria. Then the tail grabs on and, like a little syringe, it injects its DNA into the cell and starts replicating. So I have a second lesson because I hadn't read that work, which had published in 1963, that proved that many different proteins are needed to replicate DNA in addition to DNA polymerase. That was a genetics demonstration, but I had only been reading biochemical papers, so my lesson number 2 is before you do anything in science, you want to read everything you can to make sure that you're building your strategy for research on what other people already know. So this is what I spent a lot of my early career on, trying to solve a major mystery. Why were there at least 7 T4 genes that were absolutely required for replication of DNA when this virus injected its DNA in the E. coli cell? Now, because the geneticists had no idea what these things did, they just gave them numbers, 32, 41, 43, and so on. So one of these, a little later, was shown to be the T4 bacteriophage DNA polymerase, the gene 43 protein. Then there were 6 mysterious proteins needed. So what in the world could they do? Obviously, DNA replication was much more complicated than anyone had imagined. So after my 1-year postdoc, I went to Princeton, 28-year-old. We were all 28 years old as assistant professors in those days. And I focused on using the genetics to purify to purity each of those 6 unknown proteins. And I had the dream, starting in 1966, that when I had all 7 proteins, I could mix them together, and then suddenly we could replicate double-stranded DNA in a test tube. So this is a picture of my lab, 1969. Does anybody recognize me? Nobody can recognize me? I can't either, but anyway. I'm the one in the red shirt with the suspenders. And this is just a slide meant to show that science can be fun. We had a lot of fun. So I developed a strategy. As I said, I learned from my failure that you need a strategy. And so my first strategy was to develop a affinity chromatography system that I could use to isolate from bacteriophage-infected cells all the proteins that bound to DNA, with the idea that some of those mutant proteins might be among them. And it took me actually 6 months or so in Geneva, Switzerland, to develop the method, and then I went back to Princeton, and this is my first success, finding out that when I infected with a bacteriophage of any kind, there was this large band, as you see with the blue arrow, of a major protein was present, but that protein was missing whenever you infected with a mutant in the gene called 32, and that turned out to be the first discovery of the single-stranded DNA binding protein. And of course, this is my favorite protein. It got me tenure at Princeton. And what it does is it straightens out a DNA single strand and holds it in a good position with the bases exposed for the DNA polymerase. It binds cooperatively along the back of the DNA single strand. And the fact is, you could see this really nicely in an electron microscope. It's a collaboration that I did with scientist Hayo Delis, who was an electron microscopist at Cold Spring Harbor. Took a circular single-stranded DNA molecule. When you don't have 32 protein on it, it forms all these short hairpin helices, and it's all balled up in little knots, like an RNA molecule would be, due to hairpin helices of imperfect base pairs. But when you cover it with 32 protein, you get this long, extended form of the DNA. And the cooperative binding is seen by some molecules getting all the 32 protein molecules, others not getting them. So this is how the gene 32 protein helps DNA polymerase. It straightens out the strand, and we could show that the polymerase moves much faster if it's coated with 32 protein than if it's not. The fact is that we use this model system to discover a whole series of different kinds of proteins, which later were shown to be also present in all organisms, including humans. And the human version of this protein is so-called RPA protein. So I'm skipping forward. It took us a long time, but by 1975, we had collected purified preparations of all 7 proteins. When we mixed them together, we had this magical day where we could now replicate a double helical DNA template, something that I've been trying to do since 1960 when I had that dismal graduate student experience. And we needed all 7 proteins. If we leave out any one, we didn't get replication. Now, the fact is, why do you do this? The reason why you do this is so that you have a purified system with pure DNA and pure proteins, and now you could do chemistry. You could leave out one protein or only have a mixture of a couple proteins and work your way forward to what each of those proteins do. And I'm not going to go through that, but that's what we did. It took us 6 more years, us being graduate students in the lab. Just one example, the gene 43 protein turned out to form a sliding clamp for the DNA polymerase, which allows the DNA polymerase to move for hundreds of thousands of nucleotides without dissociating. Now this, when Tom and I were in graduate school, I was in a chemistry department, we all knew, my professor was a physical chemist, we all knew the enormous rapidity of collisions, so that we all thought that a cell was like a test tube with a very concentrated mixture of proteins and small molecules, and because of the rapid motions, you didn't need any organization. It just, things would collide and react. And so we viewed DNA replication as the DNA polymerase coming in, binding to the 3' adding one nucleotide, falling off, second DNA polymerase coming in, adding the next nucleotide, falling off, and so on. But it turns out life doesn't work like this. The cell is nothing like a test tube, and everything is organized. And the sliding clamp that I'm talking about, through the work of Michael Donnell and then his collaboration with John Kurian, was shown to be a ring, a donut. We actually had a graduate student working on this, and we missed it. Working in the E. coli system, Michael Donnell figured this out. So it's a very simple mechanism. The ring is sliding along the DNA, and the DNA polymerase is held to the DNA by this ring like a tether, and the DNA polymerase moves along and can't get off the DNA. It keeps on working for hundreds of thousands of nucleotides, and this is the way the replication fork looks in 2 dimensions. The top strand is the leading strand where the DNA polymerase could go forever, so it hits some horrible defect or something, but basically, you go for millions of nucleotides tethered to its ring. And as was said by Tom, the other one's got to go backwards. And that means that the DNA in the lagging strand is made as a series of short pieces that are stitched together in the end. These are so-called Okazaki pieces. Now, if anybody had told us this when we were graduate students, we would have said, this is ridiculous. This person should be thrown out of science. You never imagine that you could make one strand as a series of short fragments. It would be a mess. But in fact, that's how it works. So this is then the trombone revelation. I was giving a talk just like this somewhere in Michigan or somewhere, and all of a sudden I got this idea that this is crazy, that lagging strand DNA polymerase must be used over and over again, and I can in fact show that by manipulating my experimental system, and indeed that's the case. So the fact is that that other lagging strand is folded up around so that this trombone slide goes in and out, in and out, in and out, as each piece is made about once per second. So we now know that the same mechanism is used to replicate DNA everywhere. However, as you get to bacteria, yeast, humans, it gets more complicated in the sense that each protein has multiple subunits, is controlled by phosphorylation, all that kinds of stuff. So this was a very useful way of working in T4 and others in bacteria to really work out the mechanism. It's a shortcut to understanding humans, a very important point today when professors feel pushed by grant committees to work on human cells. And a major lesson we learned is that almost every process in the cell is driven by a complex of 10 or more proteins that work as a protein machine. That replication machine is a little like a sewing machine. To operate a machine that you know about in your experience, you have to have a series of parts, and you have to have directed motion, which is generally driven by electricity. The directed motion is also needed for protein machines that are much tinier than the ones you know that make us able to talk and listen to each other. Those are driven by ordered protein conformation changes driven by bound ATP or GTP hydrolysis in general. And this is all set up to show you this movie that was made at Cold Spring Harbor in the celebration of the 50th anniversary of the 1953 DNA discovery. So it's a wonderful illustration not only of how DNA replication works, but how life works. And that's why I'm showing it really for the latter reason. Of course, it's my favorite movie because we worked on this for so many years without understanding anything, and then finally this great understanding came from the work of many people in science. And that's why it's such a privilege to be a scientist. So let's see if we can make this work.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So that's the trombone slide in action. So some personal lessons learned. I learned a lot. First of all, if you work on a mystery that nobody understands, you'll make a major contribution because that mystery is liable to illuminate many other things. And of course, understanding how DNA replicates is related to many other issues in biology. Secondly, as I intimated, there are remarkable homologies between living things. Therefore, the shortest course to understanding human cells, even if that's all you want to understand, is to use model organisms wherever possible. We used to see people using Drosophila, worms, all kinds of things. And we have to make the politicians and the people who are trying to today guide science in inadvisable ways understand that we need to know things, that we're not going to be able to solve major human problems without understanding much more about biology through work with non-human cells. And finally, of course, we learned that nearly all cell processes will be based on elegant mechanisms too hard to predict. Now, there are lots of young students here. Important challenge for the next generation of cell biologists, biochemists, is obtaining the information needed to accurately describe the mechanism of every type of protein machine in a cell. Now, we know that there's thousands of protein machines. We know many of their parts, but we don't know how most of them work. Tom has done a beautiful job with others on understanding the ribosome as a protein machine. But there's so many protein machines that we don't understand. And these will have to be reconstituted the same way we did with DNA replication. We'll have to really do the hard chemistry to make sure we understand how they work. And there's a lot to do, a tremendous number of important challenges. I'm now going to shift to not what I learned from my own research, but what I learned from writing this textbook, which seems to go on forever. We started in 1978. These are the first 4 editions. This is the original set of authors walking across Abbey Road. The guy at the back is Jim Watson, who had the brilliant idea. He was no longer 25, of course. The same Jim Watson had a brilliant idea that in 1978 was the time to start bringing together a field called cell biology with a field called molecular biology and biochemistry. This was the release of the last edition, the 5th edition. We finally had some fun for a change. There's a lot of hard work. somebody arranged for us to be flown down to Antarctica in a C-130 Hercules by the Chilean Air Force. And here I am launching our book in January 2008 at the Chilean Air Force Base. The general had set up a podium, and I gave a little speech, and I gave him a book, and then he read another speech that somebody had written for him, and he was wondering what the heck was going on. But fortunately, there was a young doctor at the base. Of course, this was summer, but he was going to face a long, dark winter, and he grabbed the book immediately. During the winter, he was going to read the book. And at any rate, this is what it's like to write a textbook. Our textbook was successful because we all worked on everybody else's chapters. It took us a total of more than 365 12-hour days working together to produce the first edition. And in this case, this is all before word processing. This is the first draft of the development chapter written by Julian Lewis. The pen is my writing. On top of the blue pen is my writing, and I don't know who that is. Somebody else writing on top of us. So I have so many pages like this. This is why it took so long. So this is all a way of saying that we learn a lot from writing. We learn a lot from teaching. I'm a big fan of teaching as a way of learning, and also writing textbooks. And the last edition of the book, we finally came to recognize that life is incredibly complicated. Positive and negative feedback loops underlie nearly all cell chemistry, creating complex networks of interactions that will require new mathematical computational methods to decipher. I'll talk a little bit about that. And secondly, we recognize that extensive sets of scaffold proteins produce biochemical subcompartments in the cell without requiring a membrane. So this is how all biology works. When I was a student, we drew A goes to B, goes to C, goes to D, goes to E. Well, that's right, but what was left out was all these control mechanisms that make it possible to make these reactions really work in a cell. And feed forward, feed back, all kinds of mechanisms that make understanding what any set of reactions really means incredibly difficult. And I'll use a Tom Pollard example, the actin cytoskeleton, which this is a figure from our textbook, which is a vast oversimplification of all the proteins that modulate the behavior of the actin cytoskeleton. The main actin filament, a very abundant long filament in cells, is in the middle, and coming out off of it are all kinds of arrows for proteins that bind to it and modify its behavior. Coming at the bottom, off the bottom of the slide, is a famous protein myosin that hydrolyzes ATP and moves filaments. At the top left, you have the protein formin that nucleates the formation of actin from the plus end in various bundles. You have all kinds of other proteins that break up filaments, like cofilin. At any rate, this is a very complex set of interactions. This is another figure from our textbook about just the yeast cytoskeleton has all these interactions. How can we make sense of all these protein interactions? Even when we know everything, the human mind can't make sense of it. So even when we have a complete list of all the molecules, protein machines, and molecular interactions in the cell, we will not be able to understand it. Instead, life reflects what are called emergent properties that result from very complex networks of interactions. And this is why we need a lot of new ways of thinking about how to deal with this complexity. So we'll need mathematical computational models to decipher these systems. I'll give you one example, again, based on actin. So one thing we could do, we have very fast modern computers that make possible individual molecule-based simulations, so-called agent-based modeling. You can make that tens of thousands of molecules simultaneously diffuse randomly and react in a virtual space. You set up rules when they bump into each other by random motion and get so close, then they form a complex and so on. You can simulate the positions of all the molecules in steps of micro to milliseconds, and after billions of such cycles, which could take days, you can see what happens when you mix these things together. And if the emergent properties observed for the whole system of molecules closely resemble those in the cell, then one can hope to use the model to drive new experiments and make sure you actually understand mechanisms. So to use such models require a large amount of quantitative data. You really have to know all the molecules that you're dealing with, which ones bind to which. Very important, you have to know rate and the equilibrium constants for forming each partner, otherwise there's too many variables. And of course, you have to know how this same set of molecules behave in a reconstituted in vitro system that is a model system that you could then go back and forth with to try to make sure your model accounts for the facts. And of course, you also want to know that that in vitro system mimics what happens in living cells. So for this reason, you have to start with model systems that are much simpler than the entire actin cytoskeleton if you're going to model actin. Choosing those where extensive biochemical work is being done. And this example of the infection of cells by Listeria bacterium provides a model for actin-mediated movement of intracellular vesicles because this bacteria comes into the cell, gets endocytosed, then hijacks the system that normally moves small vesicles inside a eukaryotic cell and forms an actin tail that moves the bacterium around, propelling it even from cell to cell so it doesn't get exposed to the immune system. And so many people have contributed to understanding this model of Listeria. And in particular, Marie-France Carlier in France worked out an in vitro system similar to the in vitro system I described for DNA replication, but this is in vitro system for moving a bacterium, and the bacterium is replaced by a polystyrene bead plus 5 purified proteins. Secondly, and this is where Yale comes in, nearly all of the rate constant affinities for the proteins in the set have been determined by Tom Pollard and his co-workers in the USA. Actually, he was at Hopkins when he did much of this work, but you could take credit. So here's what happens when you make such a model. The actual bacteria is on the left, the model in the computer is on the right. You see the speed of motion, the jerkiness of the motion, and many other parameters look right. And the next slide is just a higher resolution view of that. In this example, the yellow actin is the actin that is polymerized with ATP bound. After it's polymerized, the tightly bound ATP is hydrolyzed to ADP, and that's the red actin. And the ADP actin is then cut by this protein cofilin, one of the 5 proteins in the set, so the tail disappears. But as you see, this is really a helpful way of sort of dissecting what all these biochemical facts mean. Now, of course, the next step is to take a next most complicated system and try to understand more and more parts of the actin cytoskeleton in this way. Of course, there's a field of computational biology that has many different approaches to trying to solve such complex issues, and there's a set of review articles in Science Magazine if you want to just start with a fairly straightforward introduction to the field. So there's a heck of a lot left to do in this field. Enormous challenge. That's why engineers, mathematicians, physicists are badly needed to work closely with biologists here. And then the second topic I'm going to talk about is scaffold proteins. When we were writing our textbook for the first 3 or so editions, we made the statement, which we thought was correct at the time, that the way you can speed up reactions in a cell is to make a membrane-bound vesicle like a lysosome or a peroxisome and concentrate the proteins that work together in that space. Now, if they're 10 times more concentrated in that space, they'll collide 10 times more rapidly and so on. That's true. But that's probably why we have peroxisomes. But it's only one of a mechanism to do that. It turns out the standard way of doing that doesn't involve a membrane. It involves something called a scaffold protein, which is a vastly under-researched area. Many things need to be done here. But basically, the idea is shown in this little cartoon from our textbook that by having a flexible protein that binds protein and RNA molecules, you confine them to a limited space. You obviously you can put them in a specialized part of the cell, which is also what happens. And you can speed up their reaction because, of course, they'll collide much more rapidly than they would otherwise. And the prototype example, again, is from Tom Pollard. The way that formin catalyzes actin polymerase. The formin is one of those actin-binding proteins that causes actin filaments to polymerize. It was at the top left of that original diagram I showed. And the formin is a red molecule, and the blue thing here is the actin filament growing in the upward direction. And what was discovered by measuring rate constants, you have to do chemistry, is that the actin filaments grow at rates that are faster than diffusion controlled. That is, faster than the rate of collision of an actin molecule with the plus end if there's nothing else going on. But of course, there is something else going on. That was what was discovered. This formin has a whisker, a scaffold, that forms a binding site for the actin and a loading zone, so that the collision of the actin molecules with the plus end are much more rapid than they would be otherwise, so the filament can grow much more efficiently than it would otherwise. More complex scaffolds were reviewed in Science magazine, as reviewed by Wendell Lim. But what life is really like is this cartoon from another review, which shows the biochemical space underneath the synapse. Highly specialized space under the synapse. Scaffold proteins in complex ways that we don't understand yet and needs a lot more biochemistry, are creating a unique biochemical compartment underneath the synapse. And this is sort of a general theme of how cells are organized. And so basically, a cell is nothing like a test tube. Cells are not only putting things in specific places inside themselves, but those places change in response to cell signaling. This is just an example. You put a phosphate on, or you put a lipid on, you put a ubiquitin on. Proteins move to different places depending on the state of the cell. And we published a nice special issue on spatial cell biology. And this series of articles even shows an article by Lucy Shapiro and colleagues that bacteria also do the same thing. Even bacteria place their proteins in specific places to enable them to be effective living cells. So my conclusion is that it will probably take most of this century to gain a true understanding of how cells and organisms work. Much more biochemistry will be needed in purified systems that reconstitute the biology, like the replication system, like the system that moves the bacteria in an actin-mediated way. Many more of those systems will need to be established. We need many more biochemists. It's a field that's been neglected in recent years. We're also going to need new quantitative methods for analyzing and understanding the enormous complexity of life's chemistry. So what I always tell people is that the first edition of our textbook, we had this statement that I think is now wrong, that as we learn more, we're bound to illuminate the incredible underlying simplicity of living systems. And in fact, they're not simple at all. We couldn't exist without this incredibly elaborate chemistry that we don't really know how to analyze yet. I mean, just because we know all the parts, there's a big emphasis now in cataloging all the genes, all the proteins, all the RNA molecules. We could see that from what we know already, that is far from being enough to reach any understanding of how life works. And so we're going to need to have new concentrated efforts. For example, how can we expect to understand a human cell if we can't really understand even the simplest bacteria? There's a bacteria called mycoplasma, which has only 500 genes or so. 500 genes, 500 RNA and protein molecules. We don't really understand that. So, why Why don't we have a major concentrated effort to get a lot of people to work together on a mycoplasma using both biochemistry and mathematical modeling so that we could actually gain the tools we're going to need to jump to more complicated systems. There's a underemphasis on this fundamental knowledge that we need in order to move forward. So in summary, where has biology been? In the past 50 years, tremendous advances have been made in our understanding of the molecular basis of life, driven largely by the development of powerful new techniques. I mean, the cost of DNA sequencing, I chaired the first committee in 1987 that recommended the Human Genome Project. At that time, the cost of DNA sequencing was $5 per base pair. We recommended that we not start large-scale sequencing of DNA until we could get the cost below 50 cents per base pair. The cost is now about a millionth of what it was when we wrote that report. So this is an example of how science feeds on science. The more you know, the more you can create new powerful methods, the faster science can go. But we're not yet done, but we can see the way to the end of a remarkable descriptive phase in cell biology, since all the molecular structures and pathways can now be deciphered, although this will still require a lot of hard work. But where's biology going? Because life's chemistry is so complex, by far the most sophisticated chemistry known, we now sort of realize that most of the most interesting attributes of life are due to these so-called emergent properties, properties that stem from very complicated networks of chemical interactions whose consequences cannot be deciphered from the details of a few individual parts alone. So we're going to need innovative new methods and approaches before we can claim to understand even the simplest living cells. So there's a tremendous amount left to do in the 6th edition of our textbook. We're going to try to do two things that we failed to do in the previous editions. One is we're going to try to make it smaller. It's too big. The more we know, it's just impossible for students. You can't get your mind around it. But secondly, we're going to try to add little examples of what we don't know. Because students, we know from talking to students that they get this idea by reading this textbook that we know everything. There's nothing left for them to do, which is just the opposite of what is true. I would estimate we know 5% about what we need to know about cells, something like that. And the second thing we're doing is we're going to have a new chapter on how we deal with complexity to make students in biology aware that many of them should think about learning some mathematics and computational skills to help us with these incredibly difficult challenges. So now I'm going to shift gears completely to a final topic, which is science diplomacy. In 2009, I suddenly got a call from the White House saying they would like me to be a science envoy. What in the world is a science envoy? Well, first of all, I learned very quickly it's an unpaid job. Second of all, it was an idea hatched by Obama's talk in Cairo in June of 2009. How are we going to start a new relationship with the Muslim world? We got off to a very bad start after 2001. And so one of his proposals that he said that he will appoint science envoys to Muslim-majority nations. And they had chosen me to be the science envoy for India and Pakistan. They chose two other people, Elias Zerhouni, who's from Algeria, anyway, the other two people they chose were obviously from Muslim countries, knew the area. I had never even thought about Indonesia, and I'm born in Chicago. Those guys were born in that region. So off I went. Now, in the end, they never were able to get me to Pakistan. Every time I was about to go, CIA officer shot somebody on a motorcycle or something, and they couldn't protect me because I'd be official representation. So I finally did my Pakistan duties by meeting a whole bunch of people in Dubai, which is not perfect by any means, and by giving talks by Skype. But I have spent a lot of time in Indonesia, and I'm about to go for the 5th time this summer because even though my appointment's over, we're still trying to see if we can make science diplomacy work. So this is Indonesia. It's the first time I went, I flew from Jakarta way over to the right side of Indonesia. It's further than across the United States. 250 million people, more than 10,000 islands. And before we went, we said, well, Americans are always going everywhere, and they're promising everything, and nothing ever happens. So let's make sure that something happens associated with this visit. And so we got the State Department to commit to supporting a event called Frontiers of Science, a kind of networking between the future leaders of science in the United States and other countries that had been pioneered by the National Academy of Sciences, initially with China, but it's also still going on with many other countries, India, Germany, so on. And so the first thing I want to show you is this result of this science envoy business. This is a picture taken at the first annual Frontiers of Science meeting. And the fellow in the front in the blue shirt is my close colleague, the president of the Indonesian Science Academy, Sanggut Marzuki, was instrumental in getting this hall to work. And so it's run by his academy and our academy. They select 35 or 40 people on each side. The model is that 6 of those people on each side are the organizing committee, and they determine what's done for 3 days. And gives young people an experience of organizing and creating something that's their own. These are sort of like a mixer because there's new grant opportunities from the U.S., the NSF peer program and the NIH peer program. So after the meeting, they could apply to be partners and get research collaboration money. My first visit, I met with the president of Indonesia, President Yudhoyono, and he said he had been a young general in the army in Kansas. He had a young son, and that son had a very good science teacher who did what we call inquiry science teaching. You know, they were solving problems, and he wanted this kind of inquiry science teacher for Indonesia. So we've been trying to help by helping send our best science teachers over to Indonesia to run workshops for their lead science teachers. And if you look at this picture way in the back, in the middle, are 2 outstanding teacher leaders from the United States who ran several workshops for the Indonesians. And of course, it's going to take continued efforts there. So 2 main themes of science diplomacy are first, promoting ties with U.S. scientists that help to strengthen science and the influence of scientists in other nations. We want Indonesian scientists to have influence with their government so their government makes good decisions about, you know, environmental issues, health issues, all kinds of issues. And these ties also build long-lasting personal relationships of trust and understanding across borders. And since we're choosing the very best 35-year-old scientists in both countries, we expect them to be future leaders in both countries and to therefore help create good relationships between Indonesia and the United States. My first experience with Frontiers of Science was when I was president of the U.S. National Academy of Sciences, and we had a meeting with the Chinese Academy, and we decided we wanted to do something like this. And I went to one of the first meetings, and the leaders on the scientific, it was a meeting in Beijing, and I met the leader on the Chinese side of their delegation. His name is Chen Zhu, or Zhu Chen, however you want to say it. Wonderful cancer biologist. Well, for the last, I think, 5 or so years, he's been the Minister of Health of China. So every time I go to China, we have a private dinner, and it's a great communication tool between our two countries. And that's the kind of thing we're trying to establish much more widely. And the second theme is helping to spread a type of inquiry science teaching that empowers citizens and promotes democracy. We want everybody everywhere around the world to be able to think like a scientist, to look for evidence, to be critical of dogma, and to try to help build a more rational world. So I'm going to end by telling you about my most recent trip. Somehow when you start being a science diplomat, even though I'm not officially one anymore, I'm still doing it. I'm still going to Indonesia. I'm going to the Third Frontiers meeting next month. I just returned from a 23-day trip to India, Egypt, and Sudan. One of the things I did was I attended the U.S.-India Frontiers of Science meeting. It was in Agra. Here's a picture taken a short while ago, actually, of the Indian and American scientists. And then after that, I went to the Sudan. I didn't even really know where the Sudan was, but it's right south of Egypt. This is the Nile. The Nile flows upward. At the top is the Aswan Dam in lower Egypt. So it flows up to the sea at Alexandria, so most of Egypt is not even shown. But much of the Nile shown here is in the Sudan. Right in the middle there, where it's the meeting of the White Nile and the Blue Nile, is the capital of Sudan called Khartoum, and that's where I was. It's right at that branch point. I don't know if you could read it, but I don't have a pointer. And here's a picture out the hotel window. It's just that the hotel was right at the meeting of the two Nile rivers, one coming from Uganda and one coming from Ethiopia. And then we're looking up towards Egypt, many thousands of miles away. Now, what was I doing there? Well, there's a movement coming out of Malaysia to help change the nature of science education in the Muslim world. And I was there because there was a workshop being organized to train teachers from that region, Sudan and other countries in North Africa, of how to teach active inquiry science to children in their respective countries. And associated with that was a workshop called The Future of Science Education, and I was the moderator. The people in the panel were from France, the UK, Jordan. The guy from Jordan is former prime minister, and the Malaysian who was most responsible for getting this whole movement going. And by the way, I'm holding a tablet I just picked up in India. You'll read about it in my last issue of Science Magazine, because I'm ending June 1st. So May 31st, our issue's got a special issue about what we learned in India about using science and technology to improve the livelihoods of poor people. And one of the things we discovered is this so-called Akash tablet, which they're producing, and will plan to give to every school kid in India, a lot of people. But basically, the fact is that it looks like a mini iPad. The next generation, I think, will be good enough to use in the United States coming in a year. And it's only going to cost $50. So I was quite excited about this, showing it to the Sudanese, but I'm also quite excited about it for the United States. So finally, we can maybe get to the point where our resources get away from these very dull textbooks that our children are plagued with, and raising all kinds of opportunities. So there's a 3-hour discussion with the audience from Sudan, and the Minister of Education was there, the woman I'm talking to here. And the funny thing is that after I left, I got an email with all these newspaper clippings. And on the front page of many of the newspapers was a picture of me, and I had no idea what it was saying. But Hannah El Samad, who's a wonderful mathematical biologist at UCSF, her native language is Arabic, and so she said what it's saying is that Bruce Alberts, since I gave a talk about inquiry science, a talk I give in many places, we don't want kids to memorize things, we want them to learn how to ask questions, solve problems, think like a scientist. We want every kid to be able to do that, and so it says, Bruce Albert says that the Sudanese curriculum in science is terrible. Well, I didn't even know what the curriculum was, but anyway, but the good part is it says also that the education minister agrees with me. So maybe we'll have some effect. But so this is part of this big dream of making the world a more rational place, the international scientists collaborating to try to help teachers teach differently, because it won't happen without active involvement of scientists in every nation. And our next meeting, in fact, is going to be in Indonesia as part of my next trip, another training session for the teachers in Indonesia. So this is my favorite quote, and it gets to the whole purpose of all this science diplomacy. There's a wonderful book written by the physicist Jacob Bronowski called Science and Human Values, published in 1956. Bronowski flew over Hiroshima and Nagasaki as a young man in the army, got incredibly depressed, asking whether science was really bad for the world, or could it be bad for the world to have science? So he spent 10 years reading and thinking. He wrote this magnificent book, and his conclusion is, of course, that science is good for the world. He said the society of scientists is simple because it has a directing purpose to explore the truth. Nevertheless, it has to solve the problem of every society, which is to find a compromise between the individual and the group. It must encourage the single scientist to be independent and the body of scientists to be tolerant. From these basic conditions, which form the prime values, there follows step by step a range of values: dissent, freedom of thought and speech, justice, honor, human dignity, and self-respect. Science has humanized our values. Men have asked for freedom, justice, and respect precisely as the scientific spirit has spread among them. And really, I think this is the major focus for me of all this science diplomacy, to try to help not only develop scientists everywhere, strengthen their ability to be effective as scientists, but strengthening their ability to affect their societies in very positive ways like this. So I'm just going to end with my last special issue on science education for Science Magazine just came out. When I finished at the academy, I was at UCSF very happy teaching students and relaxing. And I got a call from a recruiting firm asking me if I wanted to be considered for editorship of the Science Magazine. And I said, no, I don't want, no more jobs. So then I got a call saying they wanted me to do it anyway 6 months later. But the critical thing that made a difference is that I could use it to try to promote better science education, better education. And so I've tried to do that. We've had 4 special issues on science education. I'm very pleased that we've been able to put up a website with most of our science resources. We've had 2 years of prizes for the best free science education websites called SPORE, the collections at the bottom. We're still publishing the winners of 2 years of contests for the best inquiry-based college laboratories, things that can be used at Yale. Here in all these cases, there's a 2-page article in Science Magazine, plus, in this case, all the instructions you need to give that lab as an electronic supplement. A new project that I'm going to keep working on as a volunteer, Science in the Classroom, where we're trying to prepare a science article every month that's appropriate to meet the aim, help every student in the United States at least have, during their life, read one scientific article with the aim of having them understand how science works and what it is, because that's what's really critical for enabling our citizens to value the judgments of science and be able to understand what science is all about. And of course, all of our special issues, including the last one up there, and you don't need to subscribe to Science, because we want teachers to use this. This is an open access web site. So the final conclusion, my last slide, is that there are many wonderfully exciting challenges for young scientists. I've been talking to many of them today. They have so much energy and so much interest in using science in one way or the other to improve the world. I just urge all of you to be creative and intellectually ambitious. Thank you. Sure.
Bruce would be happy to answer some questions. We asked that if you want to ask a question to take a microphone. We have several Gruber Fellows around with microphones. So if you want to ask a question, please raise your hand. I'll try to right in front.
Yeah. So my question is, obviously something that's very important for like science, both in its present as well as its like future, is how it's funded. What are your thoughts on how science is currently funded, and what do you think are like would be the best way to fund science?
I'm sorry, I couldn't hear. I'm sure.
Yeah. Sorry. So obviously something that's important for both science presently as well as in the future is how science is funded. What are your thoughts on how science is currently funded, and how do you think science should be funded?
So we've been talking a lot about that today. I've been a reviewer every cycle for something called the NIH called the Young Innovator Award for people less than 7 years out of the PhD. The important part about the Young Innovator Award is that you're to do something risky, different, and you don't need any preliminary data. I wrote an editorial. So the first cycle that they had 4,500 applicants, and they gave something like 30 awards. So maybe it was 50. So I wrote an editorial 3 years ago or so saying the way to really help science in the United States is to have 500 of those, not 50. So that the common way of thinking of young scientists is I got to think of something that's risky, new, and novel. And right now, of course, as you know, the conservative system that's created by scientific review panels is forcing many young people to do the same thing as their mentors after their postdoctoral work. So I would, first of all, reserve a pot of money for starting a scientist, and second of all, I'd try to use it to encourage the best people to go in new directions. The other things I've said here, I think we've got way too much emphasis or misinterpretation of what translational medicine means. Huda Zogby was a wonderful scientist at Baylor. She wrote an editorial for Science maybe 2 months ago called The Basics of Translation. Now, she's an MD. She does beautiful work on Rett syndrome, a terrible disease children have. She got into science because she wanted to do something for these children that she was addressing as a doctor. But she made the point that trying to translate without basic understandings of biology, her analogy is it's like trying to translate Shakespeare with a first grade vocabulary. It's a wonderful editorial, which I think the leadership of NIH and many others should be paying attention to. Too many young scientists feel they have to work on human cells to get funded, and that's, as I've implied, is a very bad for making progress in the very thing that the NIH wants to do, which is solve human disease. So there are a lot of things that I think we could do better. And now that we have this stagnant budget, it should be motivating us to think carefully about how we spend our money. So the answer to your question is that we have a lot to be proud of. Indonesia, for example, has very low science funding. They have no competitive grants program. And one of the things we're trying to do is help them establish a competitive grants program. Many other countries, we just had an editorial from Japan, from Yamanaka, just won the Nobel Prize, pointing out that young scientists really don't have the opportunity in Japan that they have in the United States. So the United States is good in many ways. We've encouraged young people to be independent, but a lot of parts of our system are not working right now. And hopefully we could straighten it out before we're talking to graduate students. They say, well, I don't have to worry about it for another 8 years. So by then we hope to have it all fixed.
regarding the future of biology, you mentioned that new mathematical and computational tools are going to be needed to help us understand its increasing complexity. You also mentioned that more biochemistry is going to be needed to adequately parameterize those models. So a lot of the most useful quantitative tools we have now came from like classical biochemistry, and so do you think like the Hill equation, Michaelis-Menten, et cetera, do you think that that sort of classical period can inform scientists moving forward and trying to develop a new perspective?
What's the, I couldn't, well, the end of it, what was the end of it? I'm hard of hearing, so I thought it was only me.
So basically, like Michaelis-Menten and Hill, and they were biochemists who got these parameters, and they also developed mathematical models, which are extremely useful now, like the bread and butter. But those dated from like the 1910s, the 1930s. Do you think that there's something in that classical period that we're missing now, which can help us moving in the future? Specifically because you mentioned how mathematical models and also basic biochemistry is going to be necessary moving forward.
Well, I'm not sure. I mean, basically, you need collaborations. I don't think a person could be good at everything. I spent all my life in a code room purifying things. We need more people to be purifying proteins and setting up in vitro systems. And it'd be good if we could train people to also be on the forefront of the new kind of mathematics. I don't even know what kind of mathematics we need. Nobody does. But, so we need to get talented people at that interface. And I'm not sure what the right, but what I do know is that physicists and mathematicians and computer scientists who are very talented can waste a lot of effort if they don't deeply understand biology. So, either we have to make sure that we form partnerships that, as I said, there are a million more experiments you should do than you can do than you should do, and there's probably a billion more computational things you can do than you should do. So strategy is everything, and I think a place like Yale could really have a regular process of getting people together who have different skills and trying to make sure that what people are working on who have great computational mathematical talents is closely informed by the biology. And so it calls for new kinds of close partnerships. I'm not sure that we can expect one person to be able to do all that.
In your talk, you mentioned the Genome Project once, but you did not mention the word genomics or proteomics. Do you feel those projects are worthy for young scientists to pursue?
Sorry, I didn't. The microphone's pretty fuzzy. Did you understand the question?
Okay. It's just, so maybe I'll just shout.
Yeah.
So in your talk, you mentioned the genome sequencing project
Right.
once, and you were referencing price.
Yeah.
Sequencing. But you did not, in your future, you did not mention genomics, nor did you mention proteomics.
Okay, I got the. It's much clearer without the microphone. So why didn't I mention genomics and proteomics in my future? Because that's part of that was the first slide. We have the ability to describe. And Tom Seitz will get the structure of every one of the 30,000 proteins in the human cell. We know how to do that, and we're doing it. We're investing in it. But the mistake we're making is we're thinking that data is knowledge. Data is not understanding. So we have to there are people who are sort of hyping the idea that if we just collect enough data, enough RNA sequences, protein structures, and so on, that's going to lead to our understanding. And my major point of this whole talk really is that we need more than that. The descriptive part, which is happening, I don't need to encourage that, is not enough. we need to deeply understand and create new methods. And for example, you know, why isn't there a powerful group of people who've gotten together to try to understand a bacteria like mycoplasma? I mean, you need all kinds of skills. You need computational people, you need biochemists, you need cell biologists, we need new methods. And so if you can work out new methods, and, you know, I think it would be a major breakthrough if we could really predict the behavior of a cell with 500 gene products. But that kind of work, Lamar Smith is in the newspapers now, the head of the science committee in the House, by saying every grant has to be justified on what it's going to do for human beings. I mean, this is, you know, this, if you had to do this, you know, how am I going to explain what understanding mycoplasma is going to do for human beings? It's going to do a lot for human beings, but it's going to be 50 years from now. And if we don't do that kind of work, we're never going to get there, because just cataloging proteins is not enough. We need new, innovative approaches, and they have to be worked out in systems that are simpler than the whole complex, enormously complex human cells with this staggering complexity. Yeah.
So you can actually ask individually questions to Bruce Alberts at the reception, to which I welcome you. It's in the dining hall, to your right as you exit the auditorium, past the signs for the restrooms. So I'd like to thank Bruce Alberts.
Thank you.